Okay, to start out this video, we're going to be talking about the chemoreception of smell and taste, starting with taste. So in a taste bud, we have about 125 taste bud cells. And so this is a taste bud cell. And we're going to talk about the differences. There's basically only two differences. And it's in regards to how salt and sour depolarizes and sends a signal, or how sweet, bitter, or umami sends a signal. Two different ways. So we're going to start out with salt or sour. We have this ENaC gate, which is just basically a leak channel. And so if we eat something, this tastant of sodium comes into the cell, creates a Bwong. It's pretty simple. Affects and opens this calcium gate, introduces calcium, works on the snare proteins, right? Fuses to the vesicles, fuses the membrane. I didn't mean to draw it like that. Releasing neurotransmitters, binding and sending a signal down. H is pretty much the same thing. I'm gonna leave that. Pretty much the same way. A hydrogen ion will come in, creating a Bwong, opening this gate, doing the same exact thing, but also closes this potassium leak gate. Potassium leaving creates a more negative resting membrane potential, and so closing this gate will further the depolarization, or a more positive resting membrane potential. I think I mixed that up. I said less negative. This potassium leaking out would create a more negative, but H plus closes it, and so now we have further depolarization and even stronger signal down. Now, sweet, bitter, or umami. Sweet, carbs; bitter, you can taste with calcium or umami. Glutamate. Each one of these ligands will bind to this G protein coupled receptor and activate through the alpha subunit phospholipase C, which is a protein on here, which will send a second messenger, IP3, which will open this smooth ER full of calcium. Calcium will rush out, creating an action potential, which will open this voltage as well, so we have even more calcium, which will work on the snare proteins, fusing the vesicles to the membrane, opening them up, releasing the neurotransmitter into the synapse, sending a signal. And carbs can do it, calcium can do it, and glutamate can do it. And that is taste. Now we're going to describe smell. Here's a nose. We have the olfactory epithelium at the top of the nose, and we have this bone. It's called the cribriform plate, and we have this olfactory bulb above it that actually goes to the temporal or frontal lobe of the brain. And so when we have an odorant come in, it diffuses through this epithelium. We have this odorant, and then it binds to this dendrite, and this is how it's done. So the dendrite is blown up here. We have this GPCR, where the odorant will bind to this receptor, and the alpha unit will activate adenylyl cyclase, which produces cyclic AMP. Cyclic AMP will open this ionotropic channel, releasing sodium and calcium into the cell, which will create a Bwong, sending a signal down. It will also open up this chloride channel, which chloride will leave the cell, so making it less negative. Yeah, less negative. And so even furthering this Bwong, and it will go up through the cribriform plate. This is just cell bodies, and it synapses in this olfactory bulb, and will send a signal up to the temporal lobe or the frontal lobe, and that's how we get our smells. Okay, we're gonna move on to the eye, and specifically how light affects it. So a little anatomy, here's the eye. This right here is the cornea. We have the retina on the inside of this eye. We have the fovea, which is where light needs to focus and hit to be able to see it. We also have an optic disc right here where the optic nerves, as well as blood vessels, go in, which is a blind spot in our eye. We have this lens right here. We have our suspensory ligaments, and these purple circles are ciliary muscles. So, how light affects the eye, I'll draw in purple. If we see something, well, a couple things to understand first. A convex lens will take light and bend it in. A concave lens will receive light and bend it out. This fovea, like I said, is where the light needs to hit. Exactly. And so this is a convex lens, but distances will affect how much the light is bent. And so this needs to be able to adjust. Real quick, I'm going to explain. If we have something really close, I'm going to drop our center. And it sends light, it's going to have, the angle is steeper because it's close. And it has to bend it in a certain amount. So it hits the fovea. If we have something really far away, the angle is less steep. That's a kind of a bad drawing, but this angle is less steep, so it doesn't have to bend light as much. So it can be less convex. And I'll show you how the eye does this. All right, it's this. So if we're looking directly in an eye, we have the lens, and we have our ciliary muscles are like all around the lens. These are ciliary muscles, pretend. And then we have these suspensory ligaments. When we're seeing something up close, because of the steeper angle, we actually need a more convex lens to be able to bend the light to hit the fovea perfectly. And so what happens is these ciliary muscles will contract, meaning these suspensory ligaments will relax. They will not be tight, they'll be loose, which means the lens will be thicker. It won't be stretched out, meaning more convex. So when we're looking at something really close, our ciliary muscles contract. When we're looking at something far away, our ciliary muscles don't have to contract, so they relax, tightening up these suspensory ligaments. So the suspensory ligaments will all be stretched tight, stretching that lens, making it thinner or less convex. And so because it's less convex, it doesn't have to bend light as much to hit the fovea. Now we're going to talk about two different types of photoreceptors that we have in the eye and how they convert light into action potentials and sent to the brain. So we have two types of photoreceptors. We have rods and cones. Rods are for our dim, light, and dark. And cones are for our color. Rods are highly sensitive to light, and so that's why just dim light or a little bit of light will make them react, I mean. Cones are less sensitive. And so when there's a lot of light, like if the sun is out and shining on you, your rods will actually become what's called bleached because it's they're very sensitive and they won't really work. Where cones will still continue to work because they're less sensitive and they'll pick up color. Right here we have what are called pigmented epithelium all around our retina, and in between is our photoreceptor. We have a bunch more rods. We have about 120 million rods and 6 million cones. Most of those rods are on the peripheral of the retina. In the center of the retina is like near the fovea is more cones than rods. Okay, so now we're gonna get into how if we have dark or light, I'm actually gonna turn this a bit, how it'll affect all this. Okay, so let's start with dark. So this is our photoreceptor. We have what's called, well, the protein is rhodopsin, but it's a G protein coupled receptor we have. And inside this protein rhodopsin is 11-cis retinol. When it's dark, this rhodopsin, this 11-cis retinol will remain 11-cis retinol. When light hits it, it actually changes this double bond, this cis bond, to a trans bond. We'll go into that in a little bit. But when it's dark, it does not affect this. So this will not activate this GPCR and will not mess with this phosphodiesterase or PDE. We'll go into all that later. But here's where it starts. So this is cyclic GMP ionotropic channel, and it's non-specific. We have sodium or calcium here that can go into the cell, and it's usually open. So we have dark, not really a stimulus. Sodium or calcium will go into the cell. So calcium or sodium, this one, creating a Bourne, opening this voltage calcium gate, allowing more calcium to enter the cell. Oh, I forgot to draw my neurotransmitters. When calcium is released into the cell, it will work on the snare proteins like always, help the vesicles bind to the membrane, and release this neurotransmitter, which is glutamate. So we have, that's a check mark, we have glutamate. So important, dark in this photoreceptor means we will get glutamate released. So before we move on to the bipolar cell, we're gonna do the light scenario. Where's my eraser? So now we have light. Light hits this 11-cis retinol and changes it. That's gonna be a rough drawing. Basically like that, to a trans double bond. What that does when it turns into a trans double bond, I'm just gonna erase that because it looks ugly. It will make this G protein coupled receptor active. And so the alpha unit will activate this phosphodiesterase, PDE, and then the phosphodiesterase protein actually breaks down the cGMP. And so activate PDE, which that protein breaks down cGMP, which if we don't have cGMP, just cross that out, we will not have sodium calcium enter the cell. So we will have this way, no Blanc. So this gate will remain closed. The gate closed, no calcium will be released, meaning these vesicles will not fuse to the membrane, which means no neurotransmitter released. The neurotransmitter is glutamate. So there is, that's like a no sign. There is no glutamate. So when we have light in this photoreceptor, there is no glutamate. Okay, so now we're gonna move on to this bipolar cell. Starting with light. So no glutamate. And there's a couple different type of bipolar cells we can have. And we're gonna start with on-centered or on-centered bipolar cell. So an on-centered bipolar cell, we have in this membrane a mGlu-R6 receptor. It's a GPCR, G protein coupled receptor, that becomes active when there's glutamate. So let's keep with light since we have this up. When there's light, meaning there's no glutamate, there's nothing to bind here, so mGlu-R6 will not be active. And it will not work on this gate, TRPM1, which is usually open. And so this is a non-specific, actually it might be calcium. I'm gonna pause that and check real quick. It is not a non-specific, it is ionotropic, but it releases calcium. So calcium will come in, 2+, since it's usually open, create a Blanc, right? Open up this gate, calcium, more calcium will come in, work on these snare proteins, help the vesicles fuse to the membrane, release the neurotransmitter. And when we have neurotransmitter released, we have glutamate. So we have glutamate. On this ganglionic cell, we have an AMPA channel, which basically glutamate will bind to. Open up this gate, and AMPA is non-specific ionotropic. So cations will flow in, lots of positive charges, create a Blanc, send a signal down the ganglionic cell into the brain. So when there's light, no glutamate, which equals glutamate here, action potential. Now let's do dark again. We already messed with this. What would happen in dark? All this is gone. We have glutamate. So check. Neurotransmitters. It will bind to the mGlu-R6. Will bind to the mGlu-R6 receptor, which the alpha subunit will do its work on TRPM1, which means it will actually close it. And so no calcium will get in. So we don't have a Blanc. This gate is shut. No calcium to work on the snare protein, so these vesicles will not fuse and will not release glutamate. No glutamate, no binding to the AMPA channel. AMPA channel closes. It's getting pretty messy. No action potential. So basically what that means, on an on-centered bipolar cell, light shining on the on-centered will cause an action potential. Dark, what we just did, will not activate this on-centered bipolar cell and no action potential. Okay, so now we're going to talk about, let's clean this up a bit. Let's just erase all this. Okay. Now we're going to talk about off-centered bipolar cell. And we'll stick with dark because that's what we have here. So this off-centered bipolar cell has something a little different. Let's recreate this AMPA channel. That's closed. The off-centered bipolar cell does not have the mGluR6 receptor. It has an AMPA channel. AMPA. And so, remember, when it's dark, none of this will go on, right? So we will have a BWOG, check. Release of calcium, check. We have glutamate, GLUT for short, check. We have these neurotransmitters, will bind to AMPA. AMPA likes glutamate. When glutamate binds, it opens this channel, activates it, and the non-specific ions will come in. So Na or Ca2 plus will come in. Positive charges, right? Create a BWOG. Open up this voltage calcium gate. Calcium will flow in. Work on those snare proteins like it usually does. Helps the vesicles fuse the membranes. Releases this neurotransmitter. Check glutamate. Will bind to this AMPA. Open this gate. Positive charges, right? It's an arrow. We'll create an action potential. So, on off-centered, dark likes off-centered, basically. I don't know how else to really explain that. And so, when there's dark on an off-centered bipolar cell, we will see an action potential at the ganglionic cell. Drop a marker. Okay, so now let's switch to light. I think is our last one. Bright light. Still on the off-centered cell. Clean this up a bit. Closed. Closed. That's the AMPA receptor. So remember, light equals no glutamate. Like I've said a billion times now. So no glutamate will bind to AMPA. And so AMPA will not open, meaning there will be no BWOG, right? Or I just drew a BWOG. There will be no BWOG. And so this voltage calcium gate will not open. It will not work on those snare proteins, which will help the vesicles bind to the membrane. And so that's wrong. Let's clean that up a bit. We have our AMPA channel, right? We have no glutamate, so nothing can nothing can bind, leaving this closed, so there's no action potential. Action potential. No AP. So light on an off-centered bipolar cell will not yield an action potential in the ganglionic cell. And those are all the options and all the processes that we can have. We're going to talk a little bit more about this, and specifically how receptor fields interact with surround and center cells. So this is a very simple picture of our visual field. We have a surround and a center. And light or dark will affect each of these differently depending on the bipolar cell. And this is a little more how that's regulated, and how an action potential can be set, sent, or inhibited. So these surround cells, so important thing, these are horizontal cells. I should have wrote that. Horizontal. This is the bipolar, right? These are the photoreceptors. Photoreceptor. Okay. So, new concept, we have these horizontal cells. When the surround photoreceptor is dark, receives a dark signal, it will release glutamate, right? We learned that previously. These horizontal cells have AMPA channels that will bind. AMPA will open, basically depolarizing the cell, releasing GABA on the terminal ends of the center cell, and GABA inhibits the center cell even more. And so we will have even less, no, glutamate. If light hits the surround, we won't have glutamate come out, right? So this AMPA will stay closed, no action potential, no GABA, which will not inhibit, and so will not cause this to have even less glutamate. And depending on what hits the center determines more of what the bipolar cell will do. Back to the visual field, what light does to it. If we have an on center and light hits it, we have an action potential. If we have an off center and light hits it, we do not have an action potential. If light hits it, if it's dark, then we will have an action potential. Simply this, because this gets really confusing. So on, center, let me erase this, because we've got the point of that. On center, light equals action potential. Off center, dark equals action potential. If we have on center dark, no action potential. Off center dark, off center light, no action potential. And same with surrounds. So that's the visual field. Okay, now we're gonna start talking about the ear. More about the structure at first and how it receives mechanical signals. And so this is our ear, this is the outer ear, this is the ear canal. And so like a sound wave, or pressure wave rather, will come in and go into the, from the outer ear, the auricle, and get into the ear canal, still outer ear. The middle ear is this portion right here. This sound, or pressure wave, will come and vibrate on this tympanic membrane. And so it'll kind of like vibrate back and forth, activating the middle ear, which these are called the ossicles. And we have the malleus, incus, and stapes. And they'll mechanically, as this vibrates, move to be able to move this oval window actually. But basically, between this oval window and a lower round window, it creates more mechanical waves through this inner ear, which I'm gonna blow up and explain a little more. All right, so we're gonna talk more about the inner ear. The inner ear is basically made up of what they call a bony bony labyrinth, but we're going to mostly talk about this cochlea. So this whole thing is the cochlea. And we have 3 different layers. Well, I guess technically 4. But we have going from the base to the apex of the cochlea, which is the helicotrema. We have the scala vestibuli, and it contains perilymph. And so the pressure waves will be converted into different waves. Basically, the fluid in here in the membranes will vibrate, but the signal will travel up and then come back down in the scala tympani, which also contains perilymph. In the middle, we have this cochlear duct, which actually contains endolymph, which is more, it resembles more of a interstitial fluid, and it has full potassium, which will be important in a bit when we talk about electrical signals. But this liquid, this water basically will vibrate and basically move this basilar membrane up and down. So yeah, in purple we have this basilar membrane. And inside the basilar membrane, there's actually an organ that will help us convert these mechanical signals into electrical signals. And so I'm gonna pause it and erase all this and go into how. Real quick, I forgot on the basilar membrane how it looks. Let's see. I'm gonna draw it over here actually. So we have the base and the apex. The base, it's really small, and then it actually gets wider. So this is a short drawing, but that's how it gets bigger as it goes toward the helicotrema. And down here, that's how we hear high frequency noises. And down, or I guess higher toward the helicotrema, is how we hear low frequency. So that's the basilar membrane and how we can hear different pitches of sounds. This is how we get electrical signals from our mechanical signals earlier. This is a cross section of the cochlea. This is the scala vestibuli, scala tympani, cochlear duct, and we can see the organ of Corti in here. We have the tectorial plate. This is the basilar membrane, and we have these hair cells, and we have 4 different rows. The inner row, or I guess 2 different rows, sorry. Inner row and 3 outer rows of hair cells. These outer rows are connected to the tectorial plate. This inner row is not. So when we have a sound that goes through all the ossicles, makes the scala vestibuli, scala tympani vibrate, as well as the basilar membrane vibrate, these hair cells move up and down with the tectorial plate, and on these hair cells, these little finger things are stereocilia. Let me double check that real quick. Let me pause this. Stereocilia. And when this moves, that causes these stereocilia to bend. So, real quick too, cochlear duct has endolymph. We have out here the stria vascularis, basically how it gets potassium into the cochlear duct. So we have lots of potassium to form action potentials. So let's blow this hair cell up so we can explain something. So basilar membrane is moving. These hair cells, or the stereocilia, begin to bend. When they bend, it pulls on this tether, which is connected to a potassium gate. So it's some form of mechanical potassium gate. It opens this gate and allows potassium to come into the cell. Now, on these outer row hair cells, when potassium comes in, depolarizes this hair cell, activates this motor protein called prestin. I wrote it up here. And it's on the outer walls of the outer rows of the hair cells. When these are activated, it allows to shorten and lengthen. It's like a motor protein, this prestin, this hair cell. And so as it's moving up and down, it's also moving the tectoral plate up and down because it's connected. This causes waves to form that will mess with this inner row. This inner row is not connected to the tectoral plate, so it's not messed with. But these waves that the prestin motor proteins cause makes it so that these stereocilia will bend as well. So we're just gonna erase this prestin. This is now a inner row hair cell. So these waves cause the cilia to bend, stereocilia to bend, which we have the same mechanical gated potassium channels. And so when those bend and this potassium leaks in, it's just like any other cell. We have these vesicles containing neurotransmitters and a voltage calcium gate, 2+. Right? This potassium that came in from the endolymph will depolarize the cell, opening this calcium. Calcium will flow in, fusing the vesicles to the membrane, releasing neurotransmitters. And that's how an actual action potential and signal gets sent to the brain. These outer row are more regulatory, where this is action, signal, sending it to the brain so we can actually hear the sound that's coming in. One little thing I forgot, there's the cochlear nerve here. We'll have some form of receptors so these neurotransmitters can bind and form an action potential, or send an action potential to the brain. That's the cochlear nerve. And that is it. That is how all our special senses work.